All right, and then I will share my screen with you guys. 1 second. Oh, wait, hold on. I still had it in edit mode. No, stop sharing. Okay, I'm just gonna save that real quick. And yes, I was working on this and then I realized that there may be too many questions to get through in this time that is the Zoom. So we'll see. And I'm going to share my screen with you now that I'm no longer in editing mode. Oh, share screen, this one, share. All right, we're going to just do classic mode. And loading the game pin, go to kahoot.if. I don't know, I think Kahoots are always just kind of fun. For those of you who are just filing in, we are doing a Kahoot. If you participate in this Kahoot, that is another point of extra credit. You get a point of extra credit for being here. Thank you all so much for coming. And I am going to write down who's here. All right, Lilia, that is completely okay. Just come to the second Zoom as well and follow along with us. All right. Okay, so it
Hi,
says player
excuse me?
limit reached. Yes.
Yeah, I tried to enter, but it keep telling me the limit
Yeah,
is reached.
so I guess I've never had this many people in a review before, so I guess there is a limit to the amount of people who can be in the Kahoot. So we are going to play it. If you weren't able to get in, just follow along on the side. Keep track of the ones that you get wrong so you know what you need to study. They're pretty clearly in each, like this one's about fungus and things like that. So keep track of what you need to study and play along with us. And then in the second Zoom, I will go over any questions as well.
Okay.
So, all right, let's get this started. Okay. And start.
Can we put our answers in the chat to show that
Yeah.
that we're participating?
Yeah, you can put your answers in the chat. Right. Solution that has a higher concentration of solutes outside of the cell than inside its cell would be what kind of solution?
Can you exit on the top right where it says player limit so we could read the question?
Yes.
Thank you.
All right, yep, it is a hypertonic solution. Very, very good job, guys. Next one. Oxidation reduction reaction, the compound that gains an electron is what? Oxidized, reduced, increased, or the donor? It is reduced. So the way I remember this one, so oxidation reduction reaction, the compound that gains an electron is reduced. The way I remember this is electrons are negative. And so like if you add a negative to something, it becomes lesser. Like that's the way I remember it. Yeah, so electrons are negative, the compound that's gaining an electron gets reduced. All right, what is the final electron acceptor in anaerobic respiration? Yep, it's an inorganic molecule. Anaerobic respiration, inorganic molecule. In fermentation, it's an organic molecule, but in anaerobic respiration, it is an inorganic. An organism that derives its carbon from organic compounds and requires oxygen to grow would be called what? Heterotrophic obligate aerobe. Very good job, guys. All right, which of the following is a structure that all bacteria have? All right, ribosomes. Yes, they all have ribosomes. Some of them have those other things, but everyone has ribosomes. All right, true or false? Both gram-positive and gram-negative cells contain peptidoglycan in their cell walls. Yep, that's true. Both of them have a peptidoglycan layer. Gram-positive cells have a thicker peptidoglycan layer, while gram-negative cells have that thinner peptidoglycan layer. All right, both gram-positive and gram-negative cells contain teichoic acids in their walls. True or false? Yeah, the opposite. Yep. All right. What does it mean for DNA replication to be semi-conservative? Yep. New DNA strand is made of one parent strand and one daughter strand. All right. What kind of RNA carries individual amino acids to the ribosome for making new proteins? It is tRNA, not mRNA. It is tRNA. All right. What is a characteristic of specialized transduction? The phage incorporates itself into the host chromosome. No, a plasmid is not used. That is a different kind of bacterial genetic transfer. Which bacterial DNA transfer type requires a bacteriophage? It is transduction, not conjugation. So conjugation is that one that uses like a sex pilus and is the transfer of the F plasmid. Transduction requires a bacteriophage. So know the differences between those ones, guys. Go over transformation, transduction, and conjugation again. Rickettsia, Wolbachia, and Bordetella are all what kind of bacteria? They are gram-negative rods. So not gram-positive rods. They're all gram-negative rods. So no, you will need to know your gram stains and the shapes of your bacteria. Those are good things to remember. There are questions similar to those on the exams, if I'm remembering correctly. So if you knew one of those, you'd know the other ones, like know your bacteria shapes and things. What bacteria secretes a blue-green pigment into the surrounding media? Yes, Pseudomonas aeruginosa. You guys know that one. It's pretty distinct. All right, next one. What is the most common cause of urinary tract infections? E. coli, exactly. All right, next one. All right, Which bacterial genre are obligate intracellular parasites? There are two, and the bacteria in them are obligate intracellular parasites, which is the thing you usually associate with viruses. But nope, these bacteria do it too. Rickettsia and chlamydia. Very, very good job, guys. That is correct. All right. Which of the following is an endospore producing aerobic gram-positive rod? It is Bacillus anthracis. So Clostridium botulinum, it's not endospores. So this is a thing that gets a little bit confusing too. So you learned endotoxins and exotoxins. Those are different than endospores. They're a different thing. Endospores, and they all sound like the names sound very similar. Endospores are when an environment gets too difficult for the bacteria to live, it releases endospores, which are like little escape pods. Endotoxins and exotoxins are different things. Clostridium botulinum does produce an exotoxin, but not an endospore. All right. How does osmotic pressure work to inhibit the growth of microbes? Yep, a hypertonic environment is created. Very good job. And the bacteria cannot live in those. Right. Blank is the easiest of microbes to destroy yeah, sorry. I forgot how to read for a second. Prions. So yes, prions are the hardest to destroy. Viruses with envelopes are the easiest to destroy. So it's kind of backwards, like you'd think, oh, it has an envelope, it has an extra thing. That means it's slightly harder to destroy than viruses without envelopes. Lipids, it's a lipid envelope on a virus, and lipids are pretty easy to dissolve. Viruses without envelopes have developed other ways to evade things that would be detrimental to them. So it's viruses that are enveloped are the easiest to kill. Which of the following best describes fungi? They are eukaryotic chemoheterotrophs. So fungi are not prokaryotes, and they do not photosynthesize. So photoautotrophs, that would be plants. So fungi are not plants, and they are not bacteria. They are eukaryotic, and they are chemoheterotrophs. All right. Next one. Which medically important fungi is an obligate intracellular parasite often seen in AIDS patients? See, all of the information goes together. It is Microsporidia. So yes, know that one. It's Microsporidia, is often seen in AIDS patients. Yeah, so definitely look back over that one. All right. Which of the following is the only mycosis that is contagious and can spread from person to person? Cutaneous mycosis. Yes, very good job. those ones can be spread from person to person, so they are therefore medically important. Yeah, which herpes virus causes chickenpox and shingles? There are a million different types. There aren't, there are 4, 5, I think it's 7. HHV3, very, very good job, guys. All right. Intergenic drift is defined as small point mutations affecting H and N constantly, true or false? True. Yes. So drift, yeah, it's the small point mutations. Shift is the big, scary pandemic causing one. They rhyme, but yeah, shift is the big one, drift is the little one, and the reason we need a new flu shot each year. Which is the best, which is following is best definition of diploid cell line method of culturing animal viruses. Yep, awesome job, guys. All right. Disease contracted from a mosquito bite is an example of what kind of transmission? Biological vector transmission. Mechanical vector transmission is the one where like a fly lands on a pile of poop and then lands on your food, and it's fecal oral. Then biological vector transmission is it multiplies within the vector. All right. Which of the following is not a characteristic of innate immunity? Stores knowledge of each pathogen. Yep. All of these other things are characteristics of innate immunity. All right, which is the first cell to reach an area of injury? Neutrophils, not monocytes. It is neutrophils. Neutrophils are the ones to get there first. What is an opsonin? Yep, protein that promotes phagocytosis. And yeah, I actually think I took that question out there. Which cell is most active in cell-mediated immunity? It is T cells, not B cells. B cells are active in humoral immunity, so outside of the cell. T cells are the ones that are active in cell-mediated immunity, so stuff that's already gotten inside of the cell. All right. Which have CD4 molecule on its surface and assist B cells and T cells to react? Helper T cells, yes, very, very good job. Cytotoxic T cells are not the most helpful. They just go kill things when the helper T cells tell them to. I mean, they're helpful, but their one job is to kill things. Following is the best definition for humoral immunity. Oh, I just described this. So you guys should all get it. It's part of the immune system that uses B cells to fight microorganisms outside the cells. So make sure you're reading all of the question. This is a thing that, and this is timed, but a lot of you on your exams, there are easy answers, but you don't read the question all the way through. So the answer choice, a lot of you picked part of the immune system that uses B cells to fight off pathogens in cells. So yes, you got the B cell part right, but finish the question. It doesn't fight off things in cells, it fights off things outside of cells. So yes, this is timed, but make sure you're reading the question all the way through. All right, next one. Which of the following is the best definition of artificially acquired passive immunity? Awesome job. Yep, it's when already made antibodies are injected into you. So you're not making them yourself, so it's passive. Which of the following is not an example of a whole agent vaccine? Conjugated vaccine, yes. Both killed and live attenuated vaccines are whole agent vaccines. Conjugated vaccines are a part. Conjugated vaccines are a type of subunit vaccine. What kind of vaccine is the MMR vaccine? Live attenuated, yes. MMR vaccine is live attenuated. It is not a subunit vaccine, live attenuated. And that's why people are so worried about it sometimes. All right, which pathogen can be prevented by a subunit vaccine? Haemophilus influenza B, yes, very good job. Rubella is part of the MMR vaccine. So rubella is the R in MMR. MMR stands for measles, mumps, rubella. So rubella is prevented by MMR vaccine, which is a live attenuated vaccine. All right, the following pathogens causes a grayish pseudomembrane in the back of the throat. We've gotten to the pathogens part. Yes, this is the most recent material. All right, so now we're fully entering new territory. Bacterium diphtheria, yes. So diphtheria causes grayish pseudomembrane. That's how you see it. Like, it's very easy to tell. It's very distinct, and it is Corynebacterium diphtheriae. All right, true or false? The common cold is always a viral infection. It's true. So yeah, there can be things with cold-like symptoms caused by bacteria, but the common cold as we know it is always caused by a virus. It's caused by things in like the rhinovirus family. It's caused by things in the coronavirus family. There are millions of different kinds of common cold viruses. And that is why we can't really make a common cold catch-all vaccine, because they're all rather different, but the common cold is always a virus. All right, Gon complex is a characteristic of which disease? Tuberculosis. Yes, very good job. Not syphilis. I think anyone who answered syphilis, you might be thinking of gummas or gumas. I always forget how to pronounce it. Those are in stage 3 of syphilis, but Gon complexes are in your lungs, and those are tuberculosis. All right, which following is an acid-fast aerobic gram-positive rod with mycolic acids in their cell walls? Mycobacterium tuberculosis, yes. Yeah, and it's super slow growing, even though it's like acid fast. I don't know, that's how I remember it backwards. All right, which is line causes walking pneumonia and looks like a fried egg under a microscope. Mycoplasma pneumonia. Very good job, guys. All right, which pathogen causes bubonic plague? Yersinia pestis. Yep. Not Bordetella pertussis. That causes whooping cough. Oh no, we have 10 minutes. I think we can do this. We have like 20 questions to get through, but we'll go over more disease stuff later as well. And I'll send the Kahoot out. A vector-borne pathogen that causes symptoms, including a bullseye rash. Borrelia burgdorferi. So yes, make sure you're going over which bacteria cause which things. Borrelia burgdorferi is what the cause of Lyme's disease. All right. Gram-negative rod, vector-borne pathogen that's an obligate intracellular parasite. Or the weird type 1 up there. I don't know what that is. I was doing this at 3 a.m. last night, so. All right, Rickettsia rickettsii. Yes, very good job. And that's, Rickettsia is one of the obligate intracellular parasite. I blanked on words. All right, true or false, Plasmodium falciparum is a bacteria that causes malaria. False. Very good job. This is kind of a trick question. So Plasmodium falciparum is involved in causing malaria. However, it is a parasite. It is not a bacteria. So yes, look out for the trick questions like those. A lot of true or false. It's like half of it's true, but the other half is false. All right, which of the following is the best definition of macule? We are on to diseases of the skin. Flat reddened lesion. All right. Yeah, necrotizing fasciitis. Is it staph or is it strep? It is caused by strep skin infection. So look at those again. Staph and strep skin infections, they cause very different things. Staph aureus causes lots and lots of skin infections, but yeah. All right. Why was smallpox able to be eradicated? Yep, it has no environmental host other than humans. Same with polio, and that's why that's the one we're going for next. But an anti-vax movement made that a little bit difficult. All right. Yeah. So this is about our chickenpox vaccine. So chickenpox is a blank vaccine, while shingles is a blank vaccine. Kind of against the same virus, but they're different things. All right, so your chickenpox vaccine is a live attenuated vaccine, and your shingles vaccine is a subunit vaccine. So I don't know, best way to remember that. Kind of like shingles is comes later, and so you don't need as much of it. Whatever mnemonic works for you. What disease can be easily identified by the presence of complex spots? Measles. Yep. Very good job. Not rubella. Rubella is also called German measles, which gets confusing sometimes. But all right, which of the following bacteria does not cause bacterial meningitis? All of the above cause bacterial meningitis. Yes, they all do cause bacterial meningitis. A good rule of thumb is if there's an all of the above option, it is usually the all of the above option, unless the all of the above option is like grossly bad, but usually it is. All right, which of the following is caused by a gram-positive spore-forming rod that causes lockjaw? All right, tetanus, yes. A few more. I think we can do this. Okay. Oh yeah, where in the body does Mycobacterium leprae colonize in tuberculoid leprosy? So there are two kinds of leprosy, tuberculoid and I'm blanking on the name of the other one, but they have different places they live. It's the Schwann cells. So not the mucous membranes. The other one, it's in the skin cells. So nowhere leprosy kind of lies dormant because it stays in the body for a long time. It's a very slow progressing. Answered that one too. Yep. False. It progresses very, very slowly. Right. True or false? Both the Salk and Sabin polio vaccines were intramuscular injections. It's false. So the Sabin polio vaccine was an oral vaccine, so like dripped into your mouth, and the Salk was intramuscular. And they were at war with each other. All right. Which disease is caused by an enveloped helical ssRNA virus that causes hydrophobia?
Yeah. I got it wrong. We're doing
I think someone is unmuted. It is rabies. Rabies causes hydrophobia, which is one of the neatest things about it, I think. All right, which disease is caused by a vector-borne ssRNA virus in the Flaviviridae family? Remember, some of these are bacteria, and don't get tricked. It is Zika. So bubonic plague, none of you answered that one. That's caused by bacteria. Rocky Mountain spotted fever. Malaria is again caused by a parasite, not a virus. All right, what's a major risk factor of leaving a UTI untreated? All right, we have 1 minute and 40 seconds left. We might not get through this Kahoot, but only a couple questions left. And it's gonna kick us out, but I'm gonna be holding another, could spread to the kidneys. Very, very good job. I'm gonna be starting another Zoom, sign in with the exact same information and we'll go over any questions that you guys have. All right, why is gonorrhea difficult to diagnose in females? Yep, gonorrhea in women is highly asymptomatic, but it can cause major disease. Which stage of syphilis do chancres form? It is the primary stage, not secondary, tertiary. That's like where the bacteria gets in. So it's like the lesion. We have less than a minute. It's gonna stop me in the middle of it. Gardasil is a blank vaccine that prevents. After this question, I'm gonna stop screen sharing and we'll finish the Kahoot there. It's a subunit vaccine and it prevents HPV. Yeah. All right, I'm going to stop sharing my screen because I don't want to be kicked out mid-sentence and we're not going to be able to finish. Where's the stop sharing? There it is. All right, so definitely sign into the next Zoom. It's the same Zoom information. We're going to be going over things. All right. Recording got cut off and going to go over the last 5 questions of the Kahoot. We were so close. We were so close to finishing it. All right. So the last one was Gardasil is a blank vaccine that prevents blank. And the answer was Gardasil is a subunit vaccine that prevents HPV. All right. The next question was, okay, true or false, the small intestines have a large number of normal microbes. So true or false? Are you guys still signed in? Oh my gosh, you are. Some people are. For those of you who are answering the Kahoot, that's fair, but you can also answer in the chat. That also, all right. It is, 1 second. It's false. So the small intestines don't really have, they have a really low pH. They're low, yes, the very acidic have a low pH. Same with the stomach. So there's not a lot of normal microorganisms in there. The large intestine has a big, like a large amount of normal flora, large intestine, but the small intestine and the stomach do not. All right, the next one was, which is defined by the ingestion of a pathogen that then multiplies and then causes disease. So here, right. Maybe it would be best if I shared my screen. I'm gonna share my screen with you guys because there are question choices. Sure, all right. Yeah, I was like, I'll just copy paste them all into the chat, but that's not gonna work super well. So it is a food infection. Food infection is different from food poisoning and food intoxication. Food poisoning and food intoxication are the same thing. So food infection is when you eat a bacteria like E. coli, and then it multiplies in your gut and creates disease just because of the sheer amount of it. Food poisoning / food intoxication is when something is caused, like when a toxin is released, like in the case of botulinum. All right, which type of food poisoning / infection has the shortest incubation period? So yeah, some of these happen over the course of several days, or they can you can get infected and it'll be several days later. Some of them happen very, very quickly, and it's a terrible 24 hours. What is the chat saying? It is actually Staph aureus. So Staph aureus, you bet you didn't think Staph aureus would come back for the GI tract. It does. Staph aureus causes all kinds of human infections in every body system. Staph aureus GI tract infections, it's very, very fast. It's like incubation period is like 5 to 24 hours, usually on the lower end. And it's just causes like intestinal cramping and vomiting. But yeah, that is the shortest incubation period. What kind of hepatitis is caused by a virus in the Flaviviridae family? So yeah, lots of different kinds of hepatitis, all caused by different viruses, because hepatitis just means inflammation of the liver. So it is hepatitis C, is caused by a virus in the Flaviviridae family. And does anyone know which kind of hepatitis that we do have vaccines for? There are several different kinds, but yeah, yep. Chat, you are correct. We have vaccines for hepatitis A and hepatitis B. We do not have one for hepatitis C or D or E. All right, which GI disease is easily identified by rice water stool? And this is the last question. Cholera. Yep. Very, very good job. All right. I'm going to stop sharing my screen. Well, next, the podium. Congratulations to Erica. Very good job. And also Brina, and finally Megan. Awesome job, guys. The Kahoot, and I will share that Kahoot, and you guys can oops. You guys can use it to study. Half of the questions are from the old ones, so I would recommend studying those as well, because some of the material from your exam is going to be from old exams. Also, so Dr. Kokesh did say this in her email, and I 100% completely agree. Study your old exams. You can go back. They're the same questions, Stephanie. When will she be opening the old exams? They are open. So you can go to, if you go to your grades and you click on your exam, you should be able to see the questions, and they've been open the whole time. Like you can't retake it or redo it, and it just has like what answer is correct and what you answered. But definitely go back and study those just so you know what you got wrong on those old exams. It's also a really good refresher of the old information, because stuff that you knew very, very well for old exams, you might have, if you're like me, study it really hard for the exam and then kind of dump it out of your brain as soon as that exam is over. So since this is a bit cumulative, you will need to know stuff from those old exams. So studying, learning from your mistakes and studying the things that you got wrong is definitely a good way to study. Stephanie is asking if they're the same questions. Half of the questions on the Kahoot we just did are from some of my old Kahoots, and some of the questions from the Kahoots have been on Dr. Kokesh's quizzes and exams. So study your old exams and study the Kahoots as well. If you just want some practice questions are a really great way to study. But yes, I didn't make new questions for the old stuff, and honestly, she may not want to make questions for the old stuff. Like when you have a set of questions that's already made, making questions is hard work. And so honestly, study your old exams. There might be some questions from those on the new one. I don't know for sure. I don't get to see the exam, but if I was willing to bet, there would be some. All right, and trying to look at the questions. Will quiz questions be on the final as well? I don't know 100% for sure. I do know that some of the quiz questions have been in the exams, but yeah, so study the exams. All right, would you recommend that we watch all of the old lecture videos? I think that is rather time consuming. I think if there is a subject that you really are unsure on, like very unclear on, watch part of the old lecture video, the part that is that subject. I think it's kind of time consuming to watch the old videos. If watching videos is a really great way for you to memorize things, then go for it. Everyone has their own different study things that work for them. But I think that would be a little bit time consuming and maybe not the best way to study, unless there's a specific concept that you want to hear her explain again, in which case that would be great. Are the questions randomized between the current unit and the cumulative exam? Or are they two separate parts? I do not know. I don't get to see the exam. Think of it kind of this way. It's cumulative in the way that all of the old material has kind of been leading up to this last unit. You're learning about diseases and the specific diseases that are caused by specific things. And the whole rest of the semester, you were learning the different bacteria, the different viruses, fungi, basically the things that cause the disease. So it's kind of intermingled, because like you could get a question that's like, what is a disease, like some of the ones on the Kahoot, like what is a disease that has a symptom of lockjaw, but it is caused by a bacteria, and then it lists those bacterial factors. So it's all probably going to be mixed together, but I don't know. I don't get to see the exam. Sorry, my stomach just growled. Okay, for the food poisoning, one Staph aureus thing, is that the entero portion? Staph aureus. I don't think so. No, I'm pretty sure that's just kind of same with E. coli, like it multiplies a lot in the gut. I would have to double check the PowerPoint. I have all the PowerPoints pulled up. And I can look, and I might do that because I don't want to give you guys the wrong information. Where did it go? Is it here? Okay, yes. That was the last one I had pulled up. All right, I'm double checking. Not salmonella. Okay, Staph aureus. Okay, yeah, no, never mind. I was wrong. So it is the ingestion of the preformed exotoxin in food. So kind of the same thing as Clostridium botulinum. Staph aureus has the ability to make an exotoxin. And if you eat that preformed exotoxin, that does make sense because it is a type of food poisoning. Sorry, wait. Food intoxication, my bad. No, those are the same thing. Food infection is the different one. So yeah, Staph aureus is the ingestion of the preformed exotoxin in food. Next question is the final is 100 questions. Yes, I'm 87% sure. I should be 100% sure, but yes, the final is 100 questions. Oh, someone else confirmed. Yes. All right. Recommend watching all the old review sessions. You do not need to watch all the old review sessions. They are shorter than the lecture videos. Again, if watching videos helps you, go for it. Maybe don't watch the ones where we're doing the Kahoots because you can do the Kahoots by yourself. But yes, all of the old review videos are recorded and up on Blackboard. And the ones that are just the talking, answering questions sections probably would be a bit more helpful. Do you recommend redoing all of the study guides? Maybe, honestly. Maybe, if you have time to, I would say it's a good way to guide your studying. It's a good way to guide your learning, but at the same time, only 40% of the exam is on old material. So I would say if you have time, as a way to guide your studying, but a valuable study tool is knowing what you know versus knowing what you don't know, so that you know what you need to spend time on versus what you're like, okay, no, I got this. So it may be good to look at the old study guides as a refresher, like, oh yes, I remember these from the old units versus I don't remember this at all. Yeah, maybe look back at the old study guides. Maybe don't redo all of them unless that helps you writing things out. But yeah, it's the first 60 questions of the exam, the rest, first 60 questions, exam 4, and the rest 40 questions only final exam, correct? Nita, I'm not 100% sure what you were asking. I know this, like the split is 60-40, like 60% of the exam is on new material, like the diseases, and 40% is on old material. I guess since it's 100 questions, that could be an exact 60-40 split, but I don't know how it's going to be split up. I don't know if it's going to be like the first questions and the last questions. But yeah, 60% of the exam is on like diseases. All right, yeah. When you took the final, were the questions relatively general for the older exam material and more specific for the recent material? I'm trying to remember, but that's kind of how I feel like it was. Like, I feel like you need to understand the concepts for the old ones and how they apply to the new material. Like understanding old concept is more important than the specifics, I think, if I'm remembering correctly. Good to look over the specifics, but for studying, start big and then get smaller, so that you know the broad concepts first, and then you can narrow it down to more specific things if you have time. Do you recommend, no, okay, already did the study guide one. Is it possible to drop the cumulative exam grade and keep the current unit exam grade somehow? I do not know. I am not in charge of that portion of it. Because she drops one exam, yes? Yeah, so I don't know if you can drop the final. You're gonna study, you got this, you got it. All right, for the extra credit from attending today, will it be 1 point? Will there be another review? So the way the extra credit from the review today is working is basically if you came and I saw that you were participating and I wrote down all of your names, basically everyone in the last one, there were like 50 of you, everyone who's asking questions now. So everyone who came to the review gets 1 point of extra credit. Everyone who is actively participating and doesn't just have this on in the background gets 1 other point of extra credit. So this whole review has a capability to get 2 points of extra credit. But if you do not participate and you're just kind of having this on like to get the 1 point, then you won't get the other one. All right. She told us about some differences of pneumococcal pneumonia and another thing, and I couldn't write. Could you tell us about it? Pneumococcal pneumonia. Let me see. That would be in respiratory diseases of the respiratory tract. Let me see if I can find what you were talking about. Pneumococcal pneumonia versus what, though? Sorry, I'm going through all of the questions. I will get to all of them. But there are many. All right. Okay. So, pneumococcal pneumonia versus. Okay.
What's your name?
So there are, I think someone is unmuted. And you are allowed to unmute yourself if you're going to ask me a question, but and yeah, you can feel free to unmute if you have a question. You don't need to type it in the chat, but if you are unmuted and having a conversation with someone else, please mute your mic. So bacterial pneumonia is here. I'm going to share my screen with you guys. Sure. All right, bacterial pneumonias. So this question was okay. Told us about some differences of Shadbari. I think you asked that question. What were you asking the question specifically about? If you typed it in the chat later, I can try to find it. But like, so you have your bacterial pneumonias, yeah?
Yeah, actually, on the lecture, she talked about this pneumonia and like
The heck?
like she put something on the reverse, but I couldn't like catch up with hearing the lecture. So I just want to make sure, like, is there thing that should we know the difference between this pneumonia and other thing, or just know and read this pneumonia?
Oh, I see. Okay. So on the slide, you do have your typical pneumonias and you have your atypical pneumonias. So your atypical pneumonia is caused by mycoplasma. And that's walking pneumonia. Yes, I remember her talking about that in lecture now. So yeah, walking pneumonia is slightly different. It's caused by mycoplasma. And it's the one that looks, yeah, it's the one that looks like a little fried egg on an agar plate. You may not need to know that, but for some reason I remember like there was something about fried eggs. It may be a question, and maybe it was just a question last year. But so mycoplasma pneumonia looks like a fried egg. Hannah, I think you're unmuted.
Oh my God, I'm so sorry.
No, you're okay. You're all good. So yeah, you have your typical pneumonias, strep pneumonia, and Haemophilus influenza that causes pneumonia. Other ones caused by Staph aureus and Staph pyogenes and things. But your atypical pneumonia is walking pneumonia. Does that answer your question? All right. Will there be symptoms of Lyme disease if the tick is removed? Does the same apply to Rocky Mountain spotted fever? So it really depends. The thing about ticks is it takes them a little while to get through your several layers of skin. And so if a tick bites you, but it doesn't like really get into like your bloodstream yet, usually you can prevent disease. So like if you get bitten by a tick and you notice it within the first 24 hours, because it does take a while, which is why you should always do a tick check if you're going in the woods. If you notice it within the first 24 hours, there's a good chance that you have avoided any disease that they would spread. But yeah, if you don't see it, then there's a higher chance so basically the longer the tick is on there, the higher the chance that you will get a disease from it. Will there be photo identification questions? I do not know. I don't get to see the exam, and if there were photo identification questions, I'm not sure which ones those would be. I don't think she would have you like look at like a cold sore, like a picture of a cold sore, and be like, what disease causes this? Like, I don't personally remember any pictures from our final, like pictures like that, but I'm not 100% sure. I'm sorry. Will there be questions like realistic scenarios? Yeah, probably. So like, so-and-so is in the hospital with a burn wound and the burn is recovering and everything is going well. And then all of a sudden he gets sick again and there is a bluish green pigment on the site of the burn. What bacteria was he infected with? So there may be questions like that, like kind of explaining like this patient, like it's, yeah, I think remembering a lot of questions of like, this patient has symptoms of this, this, and this, what causes it? So definitely know your symptoms of your diseases. Because, and what's nice about the diseases is like every disease for the most part has a very specific thing. Like, oh yes, lots of diseases cause like respiratory like symptoms. Lots of diseases cause fever, but only COVID causes a loss of taste or smell, or only rabies causes you to be hydrophobic. So know the like specific ones, and those will really help you. Will new content questions be more like case studies? Oh, I think that's kind of a similar question. So yeah, they may be, but not super long, like still just a question. Is there a certain unit from the older material we should focus on more? I would say honestly, because your new materials on the diseases and you're going to need to know the causes of those diseases. Like there may be questions like some of the ones I had on the Kahoot, like which of the following is caused, like is a skin infection that's caused by a gram-positive, blah, 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 blah. So I would honestly focus probably on the bacteria and viruses unit again, but you should, and maybe on some of the first unit, like the chemistry, just because it was a while ago and a refresher would be good. How should we study the, that would make us less overwhelmed, burn out with the information. So yes, I totally get that. It is a whole lot of information. What I would say is split it up so that you're doing a little bit of it every day. Coming to this is already like a really, really good thing. You're just taking your time to like do microbiology. If you can split it up, like, okay, on these days, I'm going to work on the old material. On these days, I'm going to work on the new material. Or you could be like, I'm going to dedicate 2 hours to microbiology. It doesn't need to be the same 2 hours in a day, just 2 hours in a day. Like it doesn't need to be consecutive. First hour, I'm going to focus on the old stuff. Second hour, I'm going to focus on the new stuff. And then another thing, if you're going through the PowerPoints, one of the biggest things for me, because it is a lot of information, pick 1 to 2 pieces of information from each slide that you find important, because it can get overwhelming. There's a lot of stuff on those slides. But if you narrow yourself down, like strictly narrow yourself down to really thinking about what is the most important piece of information on this slide, then A, you have to think critically about what is the most important, and B, it definitely helps to narrow down the information. And then also knowing what you know versus what you don't know, going through those old study guides, like, oh, I remember this versus, oh, I don't remember this. So then if you remember stuff, that automatically kind of like wipes that away. You don't need to focus on it. So that narrows stuff down too. She mentioned there's going to be matching, true, false, and multiple choice will be on the exam. Will there be fill in the blanks? I'm not 100% sure. If she mentioned that there's going to be matching, true, and false, and multiple choice, then there will definitely be those 3 things. I do not know if there will be fill in the blank. I don't know if there definitely won't be, or if she forgot to mention it, but I'm not sure. I'm sorry. She'll said she will drop half the final if needed, since it's worth 2,000 points. Honestly, if Dr. Kokesh says something, then trust what she says, but I have not heard about that. All right, what is the best way to study the diseases? Best way to study the diseases is a couple every day. And maybe for like, say you're focusing on the respiratory system one day, narrow it down to these are bacterial, these are viral. So like, I like making flashcards personally. Flashcards are a bit time consuming to make though. Excuse me. So if you split it up like, oh yes, what was I saying? I lost my train of thought. But like, if you just kind of spend a bit of time with it, and then also a very, very helpful thing, on the first slide of the disease of the skin, let me see if I can find it and share it. It says exactly what you need to know for each disease. So here, I'm going to share my screen real quick. Share. So what you need to know for each disease. Yeah, for diseases, you need to know the etiology, the characteristic of the microbe. So if it's a bacteria, if it's a virus, and what kind of bacteria or virus it is. The mode of transmission. So if it's respiratory, if it's fecal oral, and its main symptoms and what's used to prevent it. So those 5 things you need to be familiar with about each disease. So that may seem like a lot, but really it's just 5 things per disease. And a lot of the things are honestly the same. Like you can split them like, oh, these are all like spread. These things are all spread via respiratory, like sorting things into piles, like to help visualize it helps me. But yeah, it's on your first slide of the skin PowerPoint. That's what you need to know for the diseases. So yeah, study those specifically things. All right. Thought she put 1 grade for the final exam in Blackboard 12. Oh, that's answering the other question. Okay. Do we have to differentiate how STIs look through pictures on the exam, or is it more descriptive? Again, I'm not sure there will be I'm not sure if there will be pictures on the exam. I am personally remembering there's more descriptions of things, but yeah, I don't think she'd give you a picture and say, identify what disease this is, but I'm not 100% sure. She skipped over many slides in class on Thursday. Was it to save time or stuff that won't be on the exam? So she study the ones that she focused on, mainly because a lot of the diseases, like there are some on the slides that she's like, no, you don't need to know this for the exam. And those are the ones she skipped over. So yeah, study the stuff she focused on first. And then if you have time, look at the stuff she skipped over. But yeah, she usually skips over stuff that's not in the exam. Thank you guys for all answering each other's questions. It is helpful. All right, can you please go over a quick overview of translations, transduction, and conjugation? Yes. So, where did it go? It is microbial metabolism. Also, I would not mind doing a 3rd Zoom if I don't get through all these questions. Just like memorize your questions because it might, the chat might delete. Or can I copy the chat? I think I can copy the chat. Save chat. Yes, I can save the chat. Ha, very nice. All right. Okay, what is translations, transduction, and conjugation in? It's under microbial genetics. That's what it is. Okay. So I'm going to share my screen again real quick, just a warning because, you know, it like messes up your guys' screens too. Okay, so conjugation, transduction, and transformation, where are they? Not transcription, that's different. Everything sounds the same. Not mutations. Okay, so transformation, transduction, and conjugation. This is the easiest slide to look at, like just at its basis form. Obviously, there's more detail on other slides, and know those details, they're good, but remember again, I do think that the old material would more so be large concepts. So transformation is the transfer of naked DNA from cell to cell. So this is just free floating pieces of DNA, maybe a cell lysed, maybe a cell died. And so its DNA is just kind of free floating, and then another bacteria picks it up. And that is one way for cell to get new DNA. Transduction is the transfer, like it's viral transfer, it requires a bacteriophage. And so that's the one where like bacteriophage, a virus, infects the bacteria, and it can either like go in, use the host cell's things to like make its own viruses, and sometimes it will accidentally, like things aren't perfect, and sometimes it'll accidentally pick up a piece of the host DNA rather than just the viral DNA. So when the cell lyses, those viruses go to other bacteria, infect other bacteria, and the piece of like cell DNA that's in some viruses then gets transferred into a new bacteria. And then conjugation is cell-to-cell contact. No, we're running out of time. We have 10 minutes. 40 minutes goes by so quickly. All right. Conjugation is cell-to-cell contact via a pili. So that's the one with the F plasmids and the HFRs. And it is basically like one cell stabs another cell and gives it genetic information. And that's its basic forms. Study the more specifics, but due to time, I'm going to go to the next questions. Okay. Yeah, can we go over the difference between food infection versus food poisoning and intoxication? So yes, food infection is when you eat something that like has E. coli on it, and the E. coli then rapidly like make more E. coli in your stomach to the point where your immune system can't fight them all off. You have E. coli in your body normally. It's just if it ends up in the wrong areas of your body, that's when it causes disease. So yeah, food infection is when you eat something and it rapidly mutates within, or not mutates, sorry, rapidly grows within you and your immune system can't fight it off. And there's just too much of a bacterial load. And then that causes symptoms. And then food poisoning / food intoxication. Food poisoning and food intoxication are the same thing. That is when you ingest a preformed exotoxin. So like in cases of Clostridium and as we learned earlier, Staph aureus. So yeah, so it's like, and it's not the bacteria itself that you ingested causing the disease, it's the exotoxin. So in food poisoning / intoxication, remember intoxication, the toxin, it's ingestion of an exotoxin, while food infection is a bacteria, you eat it and then it multiplies too much and there's too much bacteria. Will there be pictures from older units? I do not know if there will be pictures on the exams. All right. All right. Thank you, Thu. Thanks for letting me know. Okay. In general, do we have any more little tips about what will help on the actual exam taking? Like how the answer is usually all of the above. Tips to help on the actual exam taking. I mean, my main tip is always write stuff down. Use your sheet of paper that you're allowed. It has to be blank at the beginning, and then just brain dump all of your mnemonics, everything that you've studied, everything that is on your mind onto that piece of paper after you hit start, before you even look at the first question. And that always helps me, since we're not allowed to backtrack. That always helped me so much. Yeah, all of the above. Tip, honestly, read through all of the questions and all of the answers thoroughly. You don't need to rush. Just read through the questions and the answers, because there are so many times where myself and I know other people and my friends who like make silly mistakes just because they don't read through the whole answer or question or answer. Do you think there will be a lot of matching questions? I do not know the amount of matching questions in comparison to the other things. Can you tell us how to study for the exams? Want to try a new studying technique? I have 6 good study tips and I will send those out in an email. I have them written down and I can't think of them right now, but I'll send those out I'm going to send an email basically with the Kahoots and with the recordings, or maybe not the recordings. I have to send those to Dr. Kokesh because my Blackboard doesn't like me to upload them. So I will send those study tips in the email as well, and that will be sent out later tonight. What should we focus on with the new material? Those 5 things from the skin slide, like those 5 things about each disease. Can you give a brief explanation of what toxic shock syndrome is? Like where does it fall? And its basic causes? Yes, toxic shock syndrome is in, I'm pretty sure it's skin. Yeah. Is it skin? Or is it, no, it's not skin, it's nervous. Yeah, yes, that's what it was. Nervous. Ah, there it is. Okay. All right. Screen sharing. Nervous system. Share. All right. So toxic shock syndrome is caused by so nervous system, meningitis, not meningitis, not tetanus, not leprosy. Toxic shock was in the, does anyone happen to remember which section toxic shock syndrome was in? Because if it is not, I guess it's not neurologic. Stop sharing. All right. 1 second. Well, okay, but basically toxic shock syndrome, I can explain it without the slide, is essentially also caused by a toxin. It's toxic. It is when, I believe it is Staph think it is in skin. I will find it. It's when you go into shock because of the amount of okay, so remember learning about endotoxins versus exotoxins. Endotoxins are the ones that cause fever and shock. Shock is essentially a major drop in blood pressure. And that is caused by, I don't know how to multitask ever. Yes, but that is toxic shock syndrome. I cannot find the slide. It's on the slide that is on, goes into more detail, but it's essentially caused by another toxin released into the bloodstream. And it's when, like it happens a lot when someone leaves a tampon in for too long. That's a good environment for this bacteria to grow and release its toxins. And then when there's too many toxins in your blood, you go into shock, your blood pressure drops, and that is very, very dangerous.
Sorry,
Yes.
you're just answering my question. So does it just have to release one endotoxin because that's the one that causes fever? And
So no, that was a question that I remember I had last semester too. It's not just one, it's if it overwhelms. So if there's a lot of bacteria and then your immune system's fighting them off because endotoxins are released when like a bacterial cell dies and then it releases its lipid A endotoxin. Yeah, when there's too much of it and your immune system can't. So it's basically your immune system is overwhelmed and that's when you go into shock.
Okay, thank you.
Yes, of course. I lost where I was in the questions. Okay. Are there any specific pictures? I've answered that one. I don't know. Is she gonna want us to remember the diagram of COVID for the final exam? I do not know, but I do not know. I wouldn't think so, but again, I don't want to give you guys false information. Are there any extra credit points added to the final exam? Ooh, yes, that's the thing I wanted to tell all of you guys. If you have not done your course evaluations yet, do those. So there's extra credit points from coming to this and participating, but there's also extra credit offered if 70, I believe, % of the class does the course evaluations before the exam. So do your course evaluations to help out yourself and your class. Fungi, Stephanie, are you just asking about here? Let me write down the rest of the questions you guys have and we need to sign on to another Zoom. I would be happy to do so if any of you have other questions. If you don't have any more questions, you are free to go, but I will be opening another Zoom just to finish up answering questions because 40 minutes is short.
Thank you.
But yeah, of course.
Thank you for the help.
Of course, yeah. Measles and rubella. Course evaluation. Yeah, do the course evaluation for extra credit. Yeah, thank you guys so much for coming. I'm going to be opening another Zoom to answer these last final questions because I want to give you good detailed answers. So if you have any more questions, hop into another Zoom. And yeah, I'll see you guys there. Record this one real quick. Someone had a question about fungi. I forget who it was and I forget what the specific question was, but did anyone have a question about fungi? Maybe not. Someone had a question about measles, mumps, and rubella. I think it was the vaccine. Did anyone have that question? Yes, here. What was your question?
Is measles and rubella just only macular or so?
The rash caused by them?
Yeah, is it just macular rashes, or does it have any vesicular rashes on them or vesicular fluids in them?
Gotcha. Very good question. So the rashes, yeah, you will need to know what specific kinds of rashes are caused by each specific disease. And I believe, there it is. I'm just double checking. I believe it's both just a macular rash. So yeah, measles has a macular rash, and that is just flat rash, develops on your face and your like trunk, your abdomen. And then rubella is also a macular rash, so also a flat rash. They do not, measles and rubella don't have vesicular rashes. They don't have like little vesicles, fluid filled. Chickenpox does, and the fluid is contagious. But no, rubella and measles, it's just a flat macular red rash. They're in different parts, and they like different parts of the body, and they kind of present in different ways a little bit. But they're both just a macular rash. Yeah. Okay. All right. Participate in Kahoot. Yes, if you participated, you will receive 2 points. And I'm pretty sure everyone participated. So yeah. All right. MMR vaccine is given to infants. Can adults receive it? So yes. So MMR vaccine, measles, mumps, rubella, people can actually continue receiving vaccines into adulthood. We have a lot of vaccines that we get in childhood, like childhood vaccinations, like you get your chickenpox, you get your MMR when you're little. But if you need a booster as an adult, you can fully, like vaccines are safe. Adults can get boosters of vaccines. Like if someone, I know that there is a disease that just like wipes your entire immune system. So you need to get all your vaccines again as an adult, and you can fully get those. It's safe. All right. I actually I moved around a lot as a kid, and I'm pretty sure I got my second chickenpox shot. My mom's pretty sure I got it because she kept me up to date on my vaccines, but we couldn't find it because I moved around a lot when I was like getting all of those. And so I got it in high school. So yeah, childhood vaccines, you can get them as an adult. All right. Influenza A, COVID, and sinus infection. I'll do, all right. Lauren, I would email Dr. Kokesh about that specifically. I'm very sorry, and I hope you feel better, but I would email her specifically about that. Let her know, but I would do it tonight. If you do it like the day before the exam, then there would be probably no leeway. So do it as soon as possible. Oh, what was the disease associated with GON from the Kahoot? I don't recall writing that in my notes. Can anyone in the chat tell me which disease is associated with GON complexes? Tuberculosis. Yep, it is tuberculosis. So GON complexes are when you do a chest X-ray, someone who has tuberculosis will have GON complexes in their lungs. They're like little white spots there. There's a specific word for them, and I'm blanking on what that word is, but GON complexes, they are oh, okay, they're calcium deposits. That's what it is. So GON complexes are calcium deposits, and they're seen in tuberculosis, in chest X-rays in tuberculosis patients. And you do a chest X-ray to look for those GON complexes when, if the skin test comes back positive, but you're not 100% sure, because you do a skin test, but if someone has gotten vaccinated for tuberculosis, that skin test will come back positive too. So that's when you do a chest X-ray to confirm, hey, do they have immunity from it? The actual infection, or do they have, or are they getting a reaction from the actual infection, or are they getting a reaction from the vaccine? Yeah, so that's tuberculosis. Okay. Do you have any tips to remember the different types of vaccines? So for the vaccines, there's not a lot of information in that slide. There's only really like, I think 5 kinds of vaccines. I don't really have any specific tips. I think it's more so like, for me when I was studying, I was like, oh, there's not a lot of information on these slides. So like, know which 2 types are whole unit vaccines, like know that killed and live attenuated are both examples of like whole vaccines, like they have the whole microbe in them. Live attenuated, it's just like a non-infectious living version of the pathogen, while killed is just like a killed version of the pathogen, but it's still the whole pathogen. It's just killed, so it can't reproduce. Live attenuated has the ability to mutate back into its infectious form, which is why we can't give it to immunocompromised individuals. A healthy immune system has the ability to fight off, like a live attenuated vaccine before it mutates back. But an immunocompromised individual may not. So that's why we can't give a live attenuated virus to immunocompromised individuals. Maybe come up with mnemonics for the vaccines. I don't personally have any on me right now. Know that a conjugated vaccine is an example of a subunit vaccine, but not all subunit vaccines are conjugated vaccines. And then you have your toxoid vaccines, which are the ones that are made from toxins, like diphtheria and tetanus. Oh, thank you, Lauren. Yeah, see, other people definitely may have mnemonics. I do not. But yeah, Lauren, that's a good mnemonic. Remember, subunit is the B's, influenza B and Hep B. Yeah. Okay. Yes, please remember to do the course evaluation. It will, like, if most of the class does it, it's an extra credit point to you guys on the exam. And all right, I'm not seeing any more questions. I did say this one would be shorter. We're not going for the whole 40 minutes. I just wanted you guys to have the chance to ask any questions. Lauren, you can stay on for a little if you would like to. Or I know a couple of you have asked if you can stay on a little bit longer to ask about specific things. You guys can feel free to do so. And yeah, but if you don't have any further questions and you don't want to talk to me individually after, you guys are free to sign off. But yeah, thank you guys so much for coming. And you'll get those extra credit points. But yeah, thank you guys and good luck on your final. You all got this. Feel free to reach out to me all weekend long if you have any specific questions. I'm going to stop the recording.